Hello, my name is Sophia Benavente-Sayani, and I will be talking about the inhibition of nosocomial pathogens by the bioavailability of extracted botanical wide-spectrum antibiotics. In the last decade, hospitals have reported bacterial resistance to antibiotics increasing remarkably, to the point where the World Health Organization began to consider these infections caused by multi-resistant microorganisms as emerging diseases. Currently, a large part of the available antibiotics are not suitable for the treatment of these pathogens belonging to an important group of gram-negative bacteria, and a progressive increase in the incidence of strains resistant to various latest generation antibiotics has been observed, such as the antibiotic impinium, as well as strains that present decreased sensitivity to various groups of antibiotics. This has aggravated the therapeutic components that are usually used for infections caused by these pathogens. So I employed a standardized method to determine which plants have antimicrobial activity, which included employing a strand of E. coli, inoculating the bacteria with streptomarin, ampicillin, leaf extracts, and the leaf extracts infused with lemon, and recording which extracts had a noticeable impact on bacteria growth. Turns out that Piper Tosum and Coliacia esculenta were the leading extracts that resulted in a low bacteria group, and lemon proved to at times triple the effectivity of these extracts. It's evident that these methods serve as therapeutic alternatives that could potentially obtain results that can assist hospitals. So to summarize, using leaf extracts rather than antibiotics can be beneficial not only to hospitals in underdeveloped countries, but in industrialized ones as well. This is an emerging issue, especially with the rise of COVID-19. Very important that we began to turn to natural remedies that hospitals can use on the spot when necessary. Again, the objective was to determine the minimum inhibitory concentration by a plate technique as a revealer of the bacterial viability of these plants. So my investigative question was, can one find the optimal botanical extract from a tropical plant to work as a broad spectrum antibiotic without the side effects of chemical pharmaceutics in a way that lowers the number of bacteria inhibiting it? So my variables, my independent variable were the 10 types of leaf extracts I used, one batch without and one batch with lemon extracts. My dependent variable was the bacteria growth, which was dependent on the different types of leaf extracts. And my control group were the antibiotics, streptomycin and ampicillin, to compare the effectiveness of that to the leaf extract. So it was really fascinating to be able to do a litany of background research for this project, learning about gram-negative bacteria, how it affects a hospital environment, how it attacks the body, why a lot of these antibiotics have proven to be ineffective. Again, the report of a bacterial infection, medics immediately prescribe a lot of antibiotics because they do not have 100% accuracy to the pathogen that caused an infection. And it's really interesting to explore the different kinds of gram-positive and gram-negative bacteria and how they respond to the antibiotics. As you can see here, different images of the different kinds of bacteria and their different components, which all end up reflecting on how the antibiotics will respond to them. And then I explored the bacteria that I was using for this project, E. coli. Many of you may be familiar, may have heard it on the news. It's very interesting to learn about how it attacks the body, where it originates from. It was especially fascinating for me to learn about the components, as you can see here, and which different kinds of antibiotics attack different kinds of parts of the bacteria. It's also interesting to see, I guess, third-party diseases caused by E. coli, its long-term effects on the body, why it's so important for us to tackle this problem immediately. Again, like I mentioned, the different effects of these antibiotics on this particular bacteria. All right, and again, another diagram that further illustrates E. coli. All right, so more on antibiograms. Again, learning about how the antibiotics are built, what they're made out of, what makes them, widely used and widely accepted by the medical community. And then I learned about the antibiotics that I personally used in this experiment. So ampicillin, which is an antibiotic that belongs to the penicillin family, and streptococcus, which is an antibiotic that belongs to the aminoglycosides family. As you can see here, pictured were my options for antibiotics, and I ended up going with those two on the right. Again, learning about now what about the antimicrobial activity of plants, evaluating their efficiency, evaluating past studies that have said that natural remedies are indeed effective and useful. And these are the botanical extracts from tropical perennial plants that I used. While they do have complicated scientific names, they are, plants that you may have heard of before, such as guava, mango, curry, fenugreek, as well as soursop, pond, etc., etc. Again, heavily researched, heavily utilized to make sure that they dissolve very well in water. And that they could be used for the experiment. It was also very fascinating to learn about their chemical component. It was very interesting to tie in prior chemistry knowledge to these flora, learning about what they're made of and hence what makes them so effective against certain diseases. So again, just looking about their makeup, their structure, what makes them effective. And these are pictures of the plants. You are told to eat your greens, and this is why. Different textures, different colors, different shades, all play a part in their effectiveness and what makes them so great. So my materials, a long list of materials to ensure that my results were accurate, unbiased, accurate. And here are some pictures of my materials. On the left, you have a photo of right before I conducted my procedure, which I will go into in a minute, and the tea extracts, as you see over there, the leaf extracts. All right, so to create the extracts, it's kind of almost like making tea to some degree. You measure, you make sure that, it's a little more accurate than that to make sure that your results are unbiased and accurate. Here are some photos of the extracts when they were created. Very interesting process, interesting to see the different shades and colors of these extracts. And once they were all completed, they looked like this. Again, 10 leaves that were not infused with lemon and 10 leaves that were. Here I tested the teas for different minerals to see if they came out positive. Different pH levels was also very interesting, very fun to do. And now my procedure to test for effectiveness. First, starting off by clearing the workspace, making sure that everything is good to go, removing the cap off the bacteria brought, and just dipping the plastic applicator into the bottle. Then placing said brought into a agar-coated petri dish, and continuing this process for as many samples as you have. The antibiotic, as previously pictured, came in a pill-like form, and thus to replicate this process, paper discs were dipped into the extracts and placed on the broth. Then they were incubated for 48 hours, as you can see here. And then these are my results, which we will analyze later, but these, after 48 hours, you can see, significant changes in bacteria growth depending on the kind of extract. Again, process I used for agar coating the plates. All right, so the bacterial membrane has the ability to interact with MTT and thus demonstrate bacterial viability, so it is indeed effective. Out of the 10 extracts, Piperceriantosum had the highest inhibitory activity in 2 samples with the same strain of E. coli. All right, and now in graph form, we have the analysis. So it was very interesting to observe how, the different extracts reacted. They were measured in centimeters. Once they came out of the petri dishes, you could see the rings where the bacteria did not grow because the tea inhibited bacterial growth. Here it is in pie chart form. And now here are the extracts that were bioavailable, or in other words, infused with lemon. And you can see there is a significant uptick in the effectiveness. It was very visible from the get-go that when infused with lemon, these leaf extracts can be used. And they are just as, if not as effective. Right here it is in pie chart form, and then comparisons. And so you can see how in some different species, lemon extract did have a significant increase, whereas some it kind of leveled out. Towards the end, it felt almost as if lemon might have slowed down the inhibition of these leaf extracts on the bacteria. So very interesting to note and compare. Here is in a different form. Again, when hospitals take this into consideration, they may need to look at the needs of every individual patient. Here it is again. And so again, it was determined that of these 10 botanical extracts of the plants, they all presented antibacterial activity. And yeah, it was just mind-blowing to think how these can be used in hospitals worldwide to save lives. Going forward, I would like to evaluate antibacterial activity with combinations of different botanical extracts. As previously pictured, there were areas where there was no bacterial growth at all, and circles were not formed because these extracts were just so effective. It's important to assess whether there could be any type of synergy or enhancing effect with synthetic antibiotic together with the preparation of medicinal plants, not only using these plants solely, but backing them up with medication. It's also important to carry out the genetic typing of these strains to determine what the different strains have in common, really examining why some leaves were more effective than others. And yeah, continuing the research with larger populations of multi-resistant bacteria using the natural resources that plants offer. So thank you so much for listening to my presentation. And yeah, we'll be seeing plant extract cures very, very soon. Thank you.